Hello. We'll start today talking about topic 6, which actually the immune system and how the body do a lot of different kind of defenses and either specifically or non-specifically against any type of disease. So basically, we can divide the barriers of the body or the immune system into three major categories. Non-specific external barriers, and actually these are the anatomical structure like skin, secretions from the tears, saliva, or mucus. So these anatomical structure usually cover the external surface of the body and as well line body cavities like lining the digestive system, the respiratory tract, the urogenital tract. The other barrier is the non-specific barrier, and actually it is called innate immunity. That means it's a built-in immunity. So you are born with this kind of immunity. So if the external barrier, the one which is non-specific external barrier, the anatomic structure, is breached, is compromised, the innate immune response non-specifically. So some white blood cells engulf these foreign particles or destroy cells that got infected. Chemicals released by the damaged cells and the proteins released by the white blood cells can trigger two important mechanisms. We'll talk in details about these two mechanisms, fever and inflammation, in the coming slides. And this fever and inflammation are triggered by chemicals that are released by damaged body cells or proteins released from the white blood cells. The third barrier is the adaptive immunity, and actually it is called as well acquired immunity. So that's mean you acquire it or your immune got adapt through your lifetime. So basically the adaptive immunity differ significantly between each individual. And these are a type of immune response which is very specific internal defense. And in this scenario, immune cells selectively destroy the specific invading toxin or a microbe. And then the extra behavior of this defense that it can remember the invader. And this will allow a faster response if it reappears in the future. So to summarize the three defenses, you can see that once bacteria or any microbes interact with the non-specific external barrier like skin, mucous membrane, if this barrier manages to stop this microbe, it will protect the body. Once these barriers are penetrated, this will lead to the body responding by a different immune system, which is the innate immune system. And actually innate immune system, as we said, and we will talk in the next few slides about the components of the innate immune system. So basically it's a phagocytic and a specific kind of cell called the natural killer cells, and as well the two mechanisms of inflammation and fever. So what happened if this innate immune response got compromised or insufficient to combat the invader? The body is responded by a very specific immune response, which is the adaptive immune response or acquired immune response. And actually, it responded by either humoral immunity or cell-mediated immunity. What's the difference generally between humoral and cell-mediated immunity? Cell-mediated, that means cells are involved in the immune response, while humoral immunity means molecules are involved in immune response. So let's see here the anatomic and the physiological barriers of the body. You have the eyes, which actually the main defenses of the anatomical is washing off the tears, and the lysozymes that exist in the tears can kill some of the bacteria. You have in the respiratory tract, you have mucus you have ciliated epithelium, you have macrophage that reside in the alveolus, and all of them act as a barrier to first invaders. You have the genitourinary tract, you have urine that wash up a lot of pathogen. The acidity of urine as well help in killing a lot of bacteria, lysozyme as well, and the vaginal lactic acid help to make sure the vagina have less bacteria. You have the skin, which is the major anatomic barrier, and actually its secretion is antimicrobial. And of course, you have the digestive tract, which is can be function as anatomic barrier by the stomach acidity, the high acidity of the stomach, and the bile which is secreted from the liver, and as well the normal flora, the types of microorganisms, the beneficial, the harmless bacteria which live inside our gut can help to protect our body. So what about the skin and its secretion and how they protect from a pathogen? Actually, the outer surface of human skin consists of dry and dead cells. And this skin is protected by two major glands, by the sweat gland and sebaceous, which is the oily glands. And the secretion from these, and as well secretions, contain a lot of antibiotics, natural antibiotics, like the lactic acid that can be secreted from your skin. And this lactic acid provide acidity, low pH, that can inhibit a lot of the growth of bacteria and fungi. The skin as well is a host to several microbial ecosystems, including the oily sites, which like in the places of the scalp, face and trunk, the moist sites like the crook of the elbow and between toes, and as well dry sites which are all over your body. In this ecosystem, there are a lot of harmless microbes that often produce secretion that as well inhibit the growth of pathogenic microbes. The skin, you need to know that the skin always carries population of potentially harmful bacteria such as the Streptococcus and Staphylococcus. So as we said, you need to remember that the skin always carries population of Streptococcus and Staphylococcus. So this is actually the role of the skin. What about the mucous membranes? So actually mucus and the antibacterial proteins and the ciliary action defend this mucous membrane against microbes. So this mucous membrane secrete mucus, and actually it's a mucus can trap a lot of microbes that can enter your nose or your mouth. The mucus contains two major antibacterial proteins. It's the lysozyme and the defensin. Lysozymes kills bacteria by digesting their cell wall, while defensin makes holes in the bacterial plasma membranes. For cilia, which exist on the mucous membrane, they brush mucus, and they brush mucus with the microbes, with the trapped microbes, until it's either coughed or sneezed out of the body or even swallowed inside your digestive system. So what happens if it's swallowed inside your digestive system? Actually, the acidity of the stomach can, and with the acidity of the stomach, as well as the protein digestive enzymes which exist in the stomach, can help to kill these swallowed microbes. While if it's moved and go into the intestine, you have flora or bacteria in your intestine that is harmless, and these bacteria and these natural flora inside the intestine can secrete substance that destroy these invading bacteria or fungi. In females, there is a lot of acidic secretion, and mucus can protect the vagina. While the other fluid released by your body, including tears, urine, diarrhea, and the vomiting, can help as well to expel any invaders. So now we'll talk about the second barrier, which is the innate barrier, which is the built-in barrier. which is the barrier that you're born with, okay? So pathogens that penetrate these anatomic barriers, and these anatomic barriers, once compromised, for example, through a cut in the skin, a cut of the skin will lead to the entrance of a lot of pathogens. So actually, there are three non-specific types of innate immune response. Starts with the protection by white blood cells, and especially the phagocytes and the natural killer cells, NK cells. And as well, two important mechanisms is the inflammatory responses and fever. So first, for NK and phagocytic cells, these can destroy invading microbes. So phagocytes ingest invaders by a process called phagocytosis. And the types of the phagocytes involved in phagocytosis are actually three: macrophage, neutrophils, and dendritic cells. You can see that neutrophils they are active against certain bacteria, while macrophage, they are active against fungi, while the dendritic cells, they are active against any virus infected cells. And this is the three types of phagocytes involved in the innate immune response. While the natural killer cells, which is called NK cells, they provide non-specific defense against viruses. So actually they are active against virus-infected cells or even cancer cells. So make in mind and please remember that NK cells are not phagocytic. NK cells destroy virus-infected cells and cancer cells. They release proteins that make holes in the membrane of the virus-infected cells and as well cancer cells. And then through these holes, they secrete enzymes to kill the infected cells. So how do NK cells can distinguish between the virus-infected cells from healthy cells? That's an important question in order not to have unregulated innate immune response. So the NK cells have to have the capacity to differentiate between self and the non-self. So self is our healthy cells, while non-self is like the virus-infected cells and the cancer cells. So how do NK cells distinguish this difference? So the surface, to understand that, you need to know one of the major proteins, a group of proteins, set of proteins called MHC. MHC is a major histocompatibility complex, and this MHC can be displayed normally on all our normal body cells. And these MHC are very unique to each person. So we have different MHC on our cells, except for identical twins, which have same MHC protein exposed on the surface of our normal body cells. So why these MHC cells are exposed on the surface of our cells? Because these MHC proteins can mark our own body cells as self, while the virus-infected cells or even the cancer cells can lack or have different proteins exposed on the surface, that these kind of different MHC will label these virus-infected cells as a non-self to the NK. So most cancerous cells have as well missing or altered MHC protein on the surface. Therefore, they are identified by the NK cells as a non-self and can be destroyed in the same way as the virus-infected cells. So they punch holes, they make holes on the membrane, they secrete enzymes inside these cells and destroy that. So NK cells usually patrol the body, going around all over the body, and try to identify the cells that express different forms of MHC, and they recognize it as a non-self, and they encounter and start killing these cells. So let's see here the difference of the story. So you can see here a normal cell is marked by a self-marker, which is MHC. And in this case, the body immune cell recognizes this type of cell as a friend. So actually, they don't, hold on, they recognize as a self-marker, so they are happy and consider it as a friend. In other cases, either the any antigen, which is a foreign molecule exposed on the surface of any invading pathogen, they are recognized by the immune cells as a non-self, and they start to be angry and start to react. Let's see actually now what is happening. So you can see here in this animation, this is a cell here, and all of it around is NK cells, trying, because these cells looks different. So they try to eat this kind of pathogen to destroy it. So actually these are the NK cells, and you can see it have engulfed the pathogen here to the inside. See this one? It was engulfed to the inside and got destroyed. Let's see another clear animation. So in this clear animation, the NK cells, you can see this is the cancer cells, and these natural killer cells try to pour. The red dye represented the enzyme which is secreted by the NK cells inside the cancer cells, and then the cancer cells are destroyed. So actually that's live what's happening in cell. They can they did this imaging in a cell culture. So again, you can see how the cancer cells got attacked by the natural killer cells. The natural killer cells attached to the cancer cells make pores and secrete the enzymes, which is represented by the red dye, inside the cancer cells, and now the cancer cells got destroyed. Actually, it's a very cool animated image explaining the mechanism of NK cells. So what about the inflammatory response? Okay, this inflammatory response, you remember, we have inflammation and fever beside the action of the phagocytes and the NK cells. So for the inflammatory response, this inflammatory response, actually it's a very good thing for our body, because it attracts phagocytes to our injured or infected tissue. So a typical inflammatory response begins with an injury that damages cells and allow bacteria to enter to the wounded area. So the wound, plus the tissue damage, plus the microbes, invading microbes, these three factors provoke an inflammatory response. And the inflammation results in the accumulation of pus. Pus is actually thick whitish mixture of dead bacteria, plus debris, plus living and dead leukocytes, the one which acting to trying to defend the body. So here you can see that it starts with tissue damage that carries bacteria inside the wound. This tissue damage, this wounded area release very important chemicals, which is the red dots. It's called, which attract a special kind of cell, which is called mast cells. These mast cells, which is recruited to the site of injury, secrete a very important chemical, which is the histamine. And the histamine, actually, it is the major cause to provoke inflammation. This histamine, once released, this will increase the capillary permeability and the capillary blood flow, leading to the migration of the phagocytes into the site of injury. And these phagocytes actually will do its job. So it will actually ingest the bacteria and any dead cells. And this figure, it's a very simple figure describing the major steps in the inflammatory response. You need to know as well that other chemicals released by the injured cells can initiate as well. Another important step after the process of inflammation is the blood clotting. And actually the blood clotting is important because it blocks the damaged blood vessels and reducing the blood loss, and so preventing more microbes from entering the bloodstream. So what about fever? Fever actually combats a large scale infection. So it's a major systemic response more than inflammation. So you can understand now that inflammation can occur into a local site. But what if the invaders affect the whole body? Okay, so another bigger mechanism is the fever. So if invaders survive the defenses and causes serious infection, maybe systemic infection, they trigger fever, which actually this fever slows down the microbial reproduction and enhances the body's own fighting abilities. Fever is controlled actually by a very important gland in our brain. It's called hypothalamus. And the hypothalamus actually, it is the body thermostat. And usually this thermostat of our body is set around 36 or 37. So actually this is the threshold of the our body temperature, and it is controlled by the hypothalamus, the body thermostat. So certain types of bacteria, as well as phagocytic cells that respond to any infection, some kind of infection, produce very important chemicals collectively called pyrogens. And the pyrogens means fire producers. So actually pyrogens are the major cause to provoke fever inside our body, these pyrogens. So pyrogens travel in the bloodstream to the hypothalamus and raise the thermostat set point. In other words, these pyrogens make the hypothalamus aware that there is something wrong is happening inside our body. So the body responds with to increase the fat metabolism. This will generate more heat and constrict blood vessels supplying the skin, and this of course will reduce heat loss through the skin and as well will lead to shivering. And we'll understand in the next few slides how these three processes help the body to defend against the microbe. So how does fever combat infection? So actually the elevated body temperatures provoke an increase on the phagocytic cells, so increase in the activity of phagocytic cells, and as well a decrease in the reproduction of some types of bacteria, which both of them are very important as an immune defense. As well, it will increase the multiplication of cells of the adaptive immune system. What is, so it's a kind of preparing the third defense barrier, which is acquired or adaptive immune cells, by increasing the multiplication of its cell types. The fourth mechanism of fever, it is stimulate the virus infected cells, in case there is a virus infected cells, to produce a very important chemical, which is the interferon. And this interferon, which is secreted from the virus infected cells, will increase the resistance of other body cells to viral attack. So it's a very important cells to limit the propagation of the virus all over our body. So interferon as well stimulate NK cells to destroy virus infected cells. You need to make to take in mind that extremely high fever, if it's not well controlled of our body, or if it's not managed by in hospital, it can cause brain damage. So you can see here a summary of the non-specific immunity. So we have the first line of defense. You can see the mechanical barriers, the acidity of the stomach and the tears, the sneezing as well expels a lot of and saliva as well expels a lot of pathogen. For the second line of defense, you have phagocytosis, which is done by, as we said, the three different types of the immune system. You have inflammation. We talked about inflammation. We have a fever. And we remember as well, we have virogens, and this protective protein, actually, this is the main inducers of the process of fever. And as well, we have a major important cell, which is the NK cells. So this slide summarizes the components of the non-specific immunity. So you can see here, we can summarize concerning inflammation. We can summarize the cardinal signs of inflammation. So actually, it starts with, what I mean, if you got for example, like you have a cut inside your arm. So the first thing to feel is the heat. Then you have redness. Why redness? Because blood is, of course, heat, white heat, because as we said, the thermostat set point is raised, and this leads to increase in the body temperature. Why redness? Why it's redness? Because at the site of the injury, the blood is being constricted, and you can see a lot of blood cells are accumulated inside the at that site. Then you see swelling, because of course the accumulation of all these blood components will lead to the swell of the injured part. Then you feel pain, because actually the body is trying to do its job by defending. If it's not relieved these signs, you actually will lose a loss of function, of functionalism. Actually, you will not be able, if you are hurt in a finger, for example, you will not be able to use that finger. So actually, these are the 5 signs, cardinal signs of inflammation. You need to take in mind that when phased, generally the immune system, in case of the innate immune system, when faced with an invading microbes, the innate immune system simultaneously have 2 types of attacks as well, the humoral and the cell-mediated. And you need to remember that cell-mediated, that means there are cells are involved in the immune system, while humoral means there are molecules involved in the humoral immunity. Now we'll continue with the third component of the immune system, which is actually the adaptive or acquired immune system, AIR, adaptive or acquired immune system. So actually, this immune barrier starts to work if the non-specific defenses are breached. Once the non-specific systems are breached, the body mounts a highly specific and coordinated AIR, which is directed against the particular pathogen or species that have successfully colonized the body. The adaptive immune response is produced actually by the interaction among several types of leukocytes, white blood cells, the macrophage, the dendritic cells, and lymphocytes. Macrophage, as you know, macrophage, and as we already said, macrophage and dendritic cells play a role in both the innate and acquired immune response. While lymphocytes are very unique to the acquired immune response. There are 2 types of lymphocytes, the B cells and the T cells. Both B and T cells arise from stem cells in the bone marrow. Some B cells complete their development in the bone marrow, and hence they are named B cells. That's why B from bone. While others leave the bone marrow and travel along the circulation and enter the thymus, where they develop into a T cells. And these T cells got the T for thymus. Here you can see that the adaptive immune system, actually, it's concentrated around specific locations in the bone marrow, in the lymph, in the bone marrow, as you can see from the figure, in the lymph nodes. Let's have a pointer. in the bone marrow, in the lymph nodes, in the thymus, in the spleen, okay, and as well in the tonsils. These are concentrated regions of the cells of the adaptive immune system. The human body contains, as you can see, around 500 different lymph nodes. And these lymph nodes, as you can see, scattered all over the lymph vessels, and actually they contain masses of two types of cells, macrophages and lymphocytes. So the immune cells are always widely distributed in mucous membrane of airways, digestive tracts, and vagina. You can see as well here, this is the structure of one lymph node and how it consists of chambers that packaged with white blood cells, which is majorly macrophage and lymphocytes. So AIR, or the adaptive immune response, actually do three steps. So the lymphocyte recognize an invading microbes and identify it as a null self. Then the lymphocytes start to launch an attack. Then the lymphocytes, and this is a peculiar characteristic of the adaptive immune response, that it can retain a memory of the invader that allows them to fight future infection from that microbes faster and more efficient. So actually, this is the three steps of the adaptive immune response. So it needs as well three types of proteins, which are the cytokines, antibodies, and complement proteins. So actually, they coordinate with the different types of cells in the adaptive immune response to mount the defense of the acquired immunity. So what are cytokines? So cytokines, actually, they are proteins produced by macrophages and lymphocytes. They have several functions. In summary, they are used for communication between cells. They stimulate cell division in lymphocyte. Of course, you need to have more lymphocyte. And they stimulate an inflammatory response. So they add up on the inflammation which is occurring in the innate immunity. And as well, they enhance the resistance of the cell to viral infection. While antibody, it is produced exclusively by a certain type of B cells, which is, it's a type of B cell or differentiated form of a B cells, which is called plasma cells and their offsprings. So these antibody, of course, help the immune system to recognize the invading microbes and destroy them. While for the complement protein, they mostly synthesized by the liver, and they circulate in the blood, and they assist the immune system in killing invading microbes. You need to know that antibodies are exclusively part of the adaptive immune response, but some cytokines and complement protein are involved in both the innate and immune response. So how does the immune system or the adaptive immune system recognize invaders? To understand how the adaptive immune system recognizes the invaders and how this immune system or this barrier respond, you need to answer three related questions. How do lymphocytes recognize foreign cells and molecules? How can lymphocytes produce specific responses to different types of invaders? And as well, how do lymphocytes avoid mistaking body's own cells and invaders? And we'll know as well by the end of the lecture what is happening if the lymphocyte mistakes our own body cells, okay? So the adaptive immune system actually recognizes invaders' complex molecules. So it recognizes antigens. So what are antigens? Antigen comprises a lot of classes of different complexes. Antigens include proteins, polysaccharides, or glycoprotein that can trigger an adaptive immune response. Antigens are located on the surface of the invading bacteria, fungi, or other microbes. Antigens are toxins as well that can be released by bacteria dissolved in blood plasma or lymph or interstitial fluid. As well, antigens are only proteins or structure on any transplanted organs that can trigger an immune response in the recipient, that the patient who got the transplanted organ. So the antibodies, there are two kinds of molecules which is important to trigger the immune response of the adaptive immune response to antigen, which are the antibodies and an important protein expressed on T cells, which is called the T cell receptor. So lymphocytes can produce these two types of proteins that can bind to specific antigens and trigger immune response to them, the antibodies and T cell receptors. So antibodies, as we said, these antibodies are manufactured only by B cells, exclusively by B cells, and actually by the plasma cells, which is the differentiated form of B cells. And they serve two major functions in the AIR. They recognize foreign antigen and they trigger the response against the invaders, and they help to destroy invading cells or molecules that have the antigens. So this antigen antibody binding stimulate B cells to divide more, have more cells, have more plasma cells, have more antibodies to produce. While T cell receptor can recognize and bind antigen. So every T cell can produce a T cell receptor that differ from those of all other T cells. So now collectively your body will have, for example, like a million different T cells. Each one of these T cells can express on its surface a different kind of T cell receptor. And you can imagine how is the capability of this adaptive immune response to defend against million different antigens, because these T cell receptors are different on the surface of the T cells. So when an antigen binds to a T cell receptor, the T cell become activated. So it's a kind of, the T cell receptor is kind of like alarming for the T cell. Once it is sensed by an antigen or binds to the antigen, this T cell becomes activated. So what's happening if after activation of T cell? We'll see in the further slides. But you need to know that this adaptive immune system can recognize millions of different antigens, of course, owing to the different T cell receptors on all T cells, and as well the different antibodies that can recognize a lot of antigen. So the genes for the antibody can produce more than 3 million unique antibodies. That's meaning it has the capacity to defend against 3 million antigens, different antigens. That means as well that it has the capacity to defend against 3 million invaders. So here you can see the structure of the antibody. Generally, the structure consists of two heavy chains and two light chains. And the reason for the nomenclature of heavy, because it's a longer polypeptide that have larger size, bigger size, while the light chain is a smaller polypeptide that the size is smaller. So that's why it's called light chain. So you can see that each light chain bound to the heavy chain by disulfide bonds. And as well, the heterodimer here, the heterodimer, which consists of one heavy chain and one light chain, can bind to the other heterodimer by a double disulfide bonds. For each antibody, you have a variable region and a constant region. So you can see here, for the light chain, you can see the regions. For the light and heavy, you can see the region that is variable. What's the meaning of variable? That's mean that every antibody in that region have a different amino acid sequence for that region of protein. So this actually will give us an idea how the antibody are different in structure. Because in this region, this variable region of this antibody is actually the region which bind to the antigen. While the basal region of each of the light and the heavy chain for each part, it's a constant region. So to clarify this more, so if you have like million antibody, this million antibody will have this bottom region have constant region. So they are all the same amino acid sequence for all antibody. But this million antibody actually will differ in the terminal region, which is termed variable region. And why the difference is here? To enable this antibody to recognize a million different antigen. So here you can see the types of antigen which can, sorry, the types of antibody which can be secreted inside your body, either IgG, IgE, IgD, IgM, IgA. And you can see here the maximum valency of this antibody to bind is to bind to two antigen. So actually two same antigen. And as well here, the maximum capacity for that antibody is 2. It is 2. But for IgM, you can see that it have a maximum capacity of binding around 10 different epitopes or antigen, because you have 10 different antigen binding sites. While for IgA, it can bind for 4 antigen. But of course, all of these antigen have to be the same. Here as well, you can see the B cells can express antibody on its surface, and this antibody on its surface can recognize the antigen on any microbe, on the surface of any microbe. And this actually will activate as well the immune system. So the adaptive immune system as well can distinguish between self and the non-self. In this scenario, you need to make sure that B and T cells, which will be circulating inside our blood circulatory system, have to distinguish that between self and non-self. So how this occurs actually? Actually, as we said earlier, that B and T cells generally first arise in the bone marrow. So while they are immature in the bone marrow, the B and T cells first form in the bone marrow. If these immature cells can bind our own antigen, our own protein, or can recognize our own self components, they undergo apoptosis. So before leaving the bone marrow, there is like a checkpoint. Once the bone marrow found that these immature B and T cells can recognize self, they do apoptosis, which is programmed cell death. Or in other words, they are triggered to die. That's why, because in order not to pass these types of cells into the circulation, and in this case, they will provoke an immune response against self. So these immature cells, as we said, are eliminated before they mature. And the only immature cells that are allowed to develop and mature and go to the circulation will only be that recognize non-self markers or foreign reactives. Actually, they can react to foreign antigen. So again, when faced with invading microbes, the adaptive immune system simultaneously launches two types of attack, same as innate: humoral immunity and cell-mediated immunity. And you understand now that the cell-mediated immunity is controlled by the B and the T lymphocytes, while the humoral immunity are controlled mainly by the cytokines, the complement system, the antibodies, so by molecules as well. So both of the innate and the adaptive have a humoral axis and the cell-mediated axis, okay? So let's see here how the humoral immunity, humoral, we're talking about molecules, about antibodies. Mainly, humoral immunity of the adaptive immune response is actually provided by the B cells through the antibody they secrete in the bloodstream. So initially, you can see that the invading pathogen, let's have a pointer. The invading pathogen represented here by these circles, the antigen, okay, is got bind to the antibodies on the surface of the B cells. Once the B cells recognize this antigen, the B cells is selected toward this antigen and the multiply. You can see that it starts to multiply rapidly to form a lot of clone of these specific cells. These B cells start dividing and start again dividing to produce identically similar B cells to the cell originally that identified the antigen. Once they have enough amount of B cells in the circulation, they started to differentiate. So you can see that the B cells start to differentiate to two very important cells, the plasma cell and the memory cells. Actually, the main function of the plasma cell is to secrete into the circulation antibody that start defending the body against this antigen. While the memory B cells, it will return memory to got activated in a faster matter in future infection or invading by the same microbe or the same antigen. So you can see here that the plasma cells release a lot of antibody in the circulation, and you need to take in mind that this antibody all have to be of the same specificity of the original antibody on the B cells that recognize the antigen. Okay, so you can see here one of the very important clue here in the figure that you can see here. It is represented initially by 5 different B cells. All of these B cells are expressing antibody on their surface. But you can see the color coding, the different antibodies are represented by color coding. So that means once this type of B cell is triggered by this kind of antigen, this type of B cell is the only one which is allowed to divide and finally giving these plasma cells and memory cells. While the other cells, actually, what are these other cells? Actually, these other cells maybe are the memory B cells or other cells waiting for another different antigen to defend. You need to understand as well that viruses are exposed to the humoral response as well when they are outside the body cells. So once the virus is outside our own body cells, they can be attacked by the antibodies because they are recognized as well by the B cells. But viruses turn out to be safe from antibody attack when they are inside cells. So what the body do in that case? That's why every immune barrier, the innate and the adaptive, have to access the humoral and the cell mediated. So here we described how the humoral adaptive is produced. And once the virus is inside the blood, it will be attacked by the humoral response through the antibody. But if the virus succeed to get internalized inside the cell, the second axis start to work, which actually the cell-mediated adaptive response, as we'll see later. So fighting the viral infection requires the help, as we said, of the cell-mediated immune response. So let's see how the antibody actually combat these invading microbes through some mechanisms. So one of the mechanisms actually that the antibody, the specific antibody, can bind to the active site of any pathogen. In this example, like the snake venom enzyme. So it can bind to the active site of this enzyme and then render it harmless. And this process of defending is called neutralization. In other cases, the antibody can coat the microbes because it recognizes the antigen on the microbes. The process of coating of the microbes is called opsonization. And this process of coating is very helpful for macrophage to engulf the whole microbes with the coated antibody. So actually the antibody in opsonization, actually it facilitates the process of phagocytosis. When antibody bind to antigen on the surface of the invader, like these examples, it can as well interact with complement protein that is present in the blood. And these complement proteins can make as well holes in the plasma membrane of the invader and killing it. Other complement proteins make it easier for phagocytosis to ingest invaders. So actually, some other complement protein do the same function as opsonization of the antibody. So here, what about the cell-mediated immunity, which is produced by a specific T cell, which is called cytotoxic T lymphocyte? So in this stem of the adaptive immune response, the CTL actually do the same job of NK cells, if you remember from the innate immunity. So they actually attack the virus-infected cells and cancer cells with the same mechanism. So when a cell is infected by a virus, these virus-infected cells express viral protein on its surface. Each CTL, as we said, have a specific or unique T cell receptor that can sense this displayed viral antigen on the surface of the virus-infected cells. This specific T cell receptor on the CTL will bind to the viral antigen. After binding, CTL secrete proteins onto the surface of the infected cells. These proteins make holes or pores on the infected cell plasma membrane, and then CTL start to secrete enzymes that pass through the pores and kill the infected cells. So basically, it is the same mechanism of NK cells. What about the helper T cells? From the name, it implies it helps T cells, helper T cells. So actually, helper T cells enhance both the humoral and the cell-mediated response of the adaptive immune response. So, and it's a kind of a regulatory cells for the immune system. Otherwise, it will be overactivated. So B cells and the CTL cannot fight microbial invasion by themselves. They need a switch. They need a switch on for them to work. And this switch on is helper T cells. So any invader is required to be sensed first by the helper T cells. So both B cells and CTLs require the assistance of helper T cells. So helper T cells as well have T cell receptor that bind an antigen to antigen displayed on the surface of dendritic cells or macrophage. So initially, if an invader got internalized, let's take for instance a bacteria internalized and got inside our body, it got sensed by dendritic cells or macrophage. So these dendritic cells or macrophage engulf by phagocytosis these bacteria, and they display on the surface of the dendritic cells and macrophage, they display part of that bacteria as antigen. This antigen got sensed by the T cell receptor of the helper T cells. Once the helper T cells recognize this antigen, the helper T cells start to multiply rapidly, and the daughter cells of the helper T cell differentiate and release cytokines. These cytokines stimulate the cell division and the differentiation of both B cells and CTL. So again, B cells don't start to secrete antibody, and the CTL do not start to do their job by eating or killing by killing the virus-infected cells or cancer cell, unless there is a cytokines that stimulate that. So the cytokines comes from the helper T cells, and the helper T cells need to identify the antigen displayed or exposed on the surface of a dendritic cells and the macrophage. And these dendritic cells and macrophage, they are the first line who engulf this bacteria, for example, and display a part of the antigen on the surface. So here you can see a summary of all this process. So you can see here this axis, this column represent helper T cell, and this one represents cell-mediated immunity. So you can see if an invader like a virus got internalized inside our body, this virus is engulfed by dendritic or macrophage, And again, these dendritic or macrophage, which is called antigen presenting cells. So they expose part of this virus on the surface of these dendritic or macrophage. Once they are exposed on surface of this one, you can see as well, this is virus infected cell as well. Okay, so the virus have two routes, either got sensed and engulfed by dendritic cells or macrophage, or infection of the antibody cells. Soon after that, the T cell receptor of the helper T cell, you can see that helper T cells, will bind to this antigen expressed on the dendritic or macrophage, and the binding of the T cell receptor to this viral antigen will stimulate by cytokines for the CTL to bind to the infected C cell, infected cells, and got differentiated to two important cells. CTL that continue eating the infected cells, and the memory cells that keep on the memory. The helper T cells, as you can see, release cytokines that, as we said, stimulate the CTL to do the job, and they stimulate as well the formation of a memory helper cell. So you have another class which is called memory helper cell. These memory cells, the memory helper cells, and the memory cytotoxic cells, and we have memory B cells, these all will give us a stronger, more faster future immunity to the same specific virus. You can see as well that the cytotoxic T lymphocyte can release enzymes through pores on the surface of the infected cells and then destroy the cells. In this axis, you can see the humoral immunity and how the virus can be sensed by the B cells as well. With the addition of the cytokines released from the helper T cell, it will promote the B cell to differentiate into memory B cells and the plasma cells. These plasma cells will secrete antibody, which is specific as well to the original virus. So finally, you can see that we have three types of memory cells: memory B cells, memory helper T cells, memory cytotoxic T cells. And all this, as we said, all these memory cells will confer future immunity to this specific virus, but not to any other microbes. Okay, this slide, as we said, summarize the major events that occur in the adaptive immune response. And you can see as well how the helper T cell can coordinate the action of the different components of the adaptive immune response. So let's see here after the recovering of the disease, the individual become immune to a particular microbes for many years. This by the action of a memory B cells and memory T cells, which can survive for many years. And these memory B cells and T cells recognize the invader and rapidly initiate an immune response. For memory B cells, they produce clone of plasma cells, secrete the same antibody to combat the second invasion, while the memory T cells produce clones of helper T cells and cytotoxic T cells that are specific for the remembered invader. So you can see here from the figure, actually it's a very good figure, you can see the initial exposure here, and the red line represented the immune magnitude, the immune response. So you can see here at the first exposure, there is increase in immune response until finalizing and combating the invader, and the immune response will be retreat. But upon another second exposure, the magnitude of the immune response will be much more higher and in larger response magnitude to fight, and this owed to the presence of the memory B cells and T cells. A very good figure representing the course of infection of the recent SARS-CoV-2 RNA viruses or the coronaviruses. You can see initially the individual got infected, it have around 5 days of asymptomatic stage, no symptoms arise. And the onset or the symptoms start to appear from the 5th day. And here you can see the patient, you can see the progress of the antigen and the amount of the antigen represented by the blue line inside the body. As soon as, starting from day 7, the IgM, which is a type of the antibody secreted by the immune system, the adaptive immune system, start to increase by day 7, and it moves parallel with the amount of antigen of the COVID virus. So you can see it start increasing. Once it reaches its peaks, the patient begins to recover. But not only IgM helps in recovering of the patient, there is another important antibody, which is IgG. And this production begins at the day 14. And you can see it assists by the decline of IgG, by the decline of the IgM, the IgG begins production, and it provides another immunity for longer term immunity. And you can see it stay constant for a longer period up till for a longer period of time, up till to 35 days. So actually, you need to be familiar that 2 kinds of tests can be done to diagnose coronavirus. So you have a PCR test that can recognize the sequence of the nucleotides of the virus in the blood. And there is another test that can be done after recovering from the coronavirus disease to check if there is IgG present in the patient's blood. The presence of IgG in patient blood will indicate that the patient have recovered from the corona infection. So here you can see a summary that differentiate the innate and the immune and adaptive. Mainly the specificity of these in the innate is non-specific, while for the adaptive it's very specific. And so you can see here that the innate immune response is provoked from pathogen-associated molecular patterns, which actually the antigen, which we have said, displayed on the surface of the invaders. While for the adaptive, it's very specific for antigen and the microbial invaders. The diversity, of course, for the non-specific, for the innate is limited. The diversity is very high for the adaptive. There is no memory in the innate. The self, of course, both of them have no self-reactivity. The response to first invasion of any particular antigen is immediate for the innate because it's first line of defense, but it is late to 2 weeks until the body got activated and B and T cells got divide and secrete the antibodies. But for the second invasion, actually there is no difference in the innate response between the first and second invasion, but the difference is great for the adaptive response because there are enhanced response in the second invasion of the same antigen. For the component, as we said, we have humoral and cell-mediated. For the component, the anatomic and physiological barrier, of course, for the innate skin, mucosa, and the pH of the stomach, all of this we have discussed in the innate. While for the adaptive, you have the lymph node, spleen, mucosal, some mucosal associated lymphoid tissue. For the blood protein, you have the complement system here, you have the antibody for the adaptive. For the cells, of course, the major 2 cells are the natural killer cells and the phagocytes. While for the adaptive, you have the B lymphocytes, which with its subtypes, the plasma cells and the mammary cells, and you have T lymphocytes with its subtypes, the T helper and cytotoxic T lymphocyte. And this table actually summarizes what we have said and all the functions of the different types of the immune system. So actually all these table have been said in every previous slide. It's kind of like a summary for all the function of the different cell types, and it's a very important table. So what happens if something wrong for the immune system? Allergies are misdirected actually immune response. So allergies are immune reaction to harmless substance. So common allergies include those in pollen spores, bee, wasps, food like milk, eggs, fish, wheat, tree nuts, or peanuts. So an allergic reaction begins when allergy causing antigen called allergen it entered the body and bind to the allergy antibodies, which is IgE, antibodies on a special type of B cell. So you can see here, initially, the body is exposed to a bowling green, for example. This bowling green, for some reason, it's not known until up till now, it causes the plasma B cells to be differentiated into plasma cells. And hence it start, these plasma cells produce allergy antibodies, the red ones. This allergy antibody binds to a mast cell. Once it bound to the mast cell, this mast cell secrete histamine. Okay, the re-exposure of this antigen will produce more antibodies on the mast cell, and then the binding of these pollen grains stimulate the mast cells, of course, to secrete histamine. And if you remember from the beginning of the lecture, histamine will trigger the inflammatory response of our body. Researchers are not sure why immune system causes an allergic reaction when a normally harmless foreign substance enters the body. So allergies have a genetic component. So actually, a lot of studies says that it is transmitted genetically. This means that parents can pass them down to their children. So an autoimmune disease, actually, it is the immune response against the body's own molecules. So the immune system reacts against our own cells. Our immune system rarely do mistakes. And due to some reason, mostly unknown, antibodies are produced as anti-self. So the antibody reacts with our cells. The result is an autoimmune disease in which the immune system attacks on body cells. An autoimmune disease include a lot of examples. That type 1 diabetes, and actually the immune cells attack insulin secreting cells of pancreas, while rheumatoid arthritis affect the joints, and the multiple sclerosis affect central nervous system. That means the autoantibody or anti-self antibody attack the central nervous system, leading to degeneration of neurons, as well as systemic lupus affect almost any part of the body, and the Crohn's disease, which mainly affect the digestive system. Unfortunately, there are no cure up till now for autoimmune disease. As well, immunodeficiency diseases in which body cannot mount an effective immune response. About 1 in 500 people has some type of genetic acquired immunodeficiency. And this acquired immunodeficiency may arise for several causes, including radiation, chemotherapy, immune suppressive drugs, diabetes, leukemia, and the pathogenic microbes. One of the major immune deficiency syndrome is a severe combined immune deficiency, SCID. It is inherited disorder affecting almost 1 from 660,000 children is born actually with SCID. It is characterized by few or no immune cells are formed, so it's an immune deficiency. As well as acquired immunodeficiency disease, or syndrome, which is the AIDS. And it's caused by the human immunodeficiency virus, HIV. And this virus actually infect and destroy helper T cells, which regulate and activate the immune system, as we discussed in the lecture. And these helper T cells stimulate both the cell-mediated and humoral immune response. The HIV enters a helper T cells and hijacks its machinery and the metabolic machinery, forcing it to make more enzymes. The life expectancy for untreated AIDS patients or victims is maximum by 2 years. Several drugs can now slow down the replication of HIV and thereby slow the progress of AIDS. And this concludes our lecture for the defense against disease. In next lecture, we will discuss the endocrine system.